All right, perfect. Well, thank you, Billy, for the introduction and greetings all. My name is Omar Rivera. Sorry for the technical issue there. So today I will be presenting the current project that I'm lead on, and the title is Evaluating Analytical Method Performance for Breath Analysis and RST to assess lab performance. So before I proceed, I want to thank all of my work group in general. Without them pretty much just performing all of the work, none of this progress would be possible. So as part of our disclaimer, it's very similar to what Eric and Victoria have discussed so far. So it's similar to everything that they presented so far. So I will skip on to the next slide. So in general, as part of our project scope, chemical detection variability amongst labs can impact findings and conclusions. We wanted to develop a process or scoring scheme that would allow us or allow labs to evaluate analytical methods and use it as an important tool to reliably detect chemicals. Usually will require careful consideration of the analytical method parameters. The problem we're trying to solve is the unclarity if a biomarker found in one study could be found in another, and if the analytical methods used were robust enough to detect such. Our aim is to develop methods which will bring consistency among studies and detection methods. So as part of our proposed solution, our intended approach for the breath chemical universe is to predict if a method can detect a chemical and then apply that particular procedure to all the relevant chemicals in your analysis. In essence, a question that we usually ask ourselves is how effective is the method at detecting all these relevant chemicals? We anticipate to address this problem by developing a GCMS retention and response predictions for relevant volatile chemicals. Our focus will be breath and TB biomarkers that have been reported so far. We intend to examine a potential universe of relevant breath chemicals, and in doing so, we expect to evaluate method performance and compare capabilities between the methods. So as part of our schematic, or in general, our experimental approach is our intent. We intend to develop this breath universe through the search of public databases and literature related with breath compounds. So sampling will entail collecting a series of VOC standard compounds that you can normally purchase commercially. So we've employed a non-targeted approach when selecting our GCMS methods to perform all of our experiments. Our plan is also to evaluate our GCMS methods based on modeling response and retention prediction of the analytes of the breath universe. Our expectation is that the models would allow us to determine the detectable space in the universe. And to further enhance the applicability of the universe, we plan on employing all of Dr. Eric Miller's results from his high resolution and low resolution GCMS experimentation. And our intention is to then develop a scorecard, which is expected to show all of the boundaries of the chosen analytical methods. So we intend to collect all relevant data to provide those developed methods to collaborating laboratories so they could perform all of the experiments that we have designed. And we can then evaluate all those inner lab scores for comparison purpose. So the results generated are expected to yield a regulatory science tool. So as part of our methodology in general, and currently as part of our ongoing work, we have been performing our experiments with headspace gas chromatography. So our focus is on understanding and probing what determines the detectability in the headspace gas chromatography technique, in headspace gas chromatography from a point of view of the retention on the column, as well as ionization and detection. Our first step of the project from our developed chemical universe of breath-related volatile compounds was to create a list of reference standards based on common functional groups as well as their cumulative representation. From there, we were able to select the analytes that represented a broad cross-section of different physical-chemical parameters as well as their functional groups. So one of the variables, the first variable that's selected in our ongoing work, as I mentioned in the previous slide, it's retention on column. And if an analyte is within a particular method retention range, a very common technique to determine an actual instrument performance is using an alkane ladder, as you could see from the figure on the bottom left. So to calibrate the retention time to retention index and retention index prediction for those analytes, we've already have established several different methods within our laboratory. And a parameter to also consider is the partition coefficients of the analytes that are usually found within your retention range, as shown in the bottom right figure. So the other sort of parameter we considered or are currently focusing on our ongoing efforts is the detection capabilities. So in general, one thing that we should note is that an analyte must be above the detection, above the instrument detection capabilities, or above the limits of detection of for that design method. So as part of our experimental design, one of the most important factors we considered for detection was the ionization efficiency. So when we think about ionization, we always think about the collisions that occur inside of the particular mass chamber. But in our end, to model the detectability, we have implemented the concept known as electron impact cross-ionization. This method, briefly, in general, just employs a mathematical model to correlate experimental retention properties to a group of descriptors that in general, as shown here, employs that mathematical model to correlate those experimental retention properties to a group of those descriptors that encode a structural role or a physical chemical property of the compounds under analysis. So the theoretical methods that we're using are structural-based computational approaches with the generation of many theoretical descriptors that subsequently reduce to a more manageable number of statistical tools. So these particular descriptors that you see here in the bottom right are then used as variables in a non-linear or linear model to sort of establish a predictive relationship, as you could see on the left-hand side, between the retention and of the compound and its actual properties in general. So how does this all relate to what we're doing with our Gates and Owlstone collaboration? So with the concept that I've described so far, our intention with our ongoing efforts to apply these for the development of a chemical universe and its applications to TB studies. To determine the breath reference standard selection, we plan on using an alkane ladder that's built in-house. And detection response ladder is chosen from a list of relevant chemical standards that we've selected in our laboratory. As far as a construction of our breath chemical universe, we've gathered a lot of information from a variety of sources. So we chose all those chemicals with a bracket of a range of possible values for each chemical, physical chemical parameter within a bracket of minimum, maximum, and 3 medial values for each particular parameter. One of our testing plans for our project, or as part of our intended testing plan, We're considering an approach reported by the best practices for non-targeted analysis. So in essence, it's BP4NTA. So our idea with this approach is that we want to use very few chemicals to respond to predict the response characteristics of a larger group of chemicals. So our intentions is to use a confusion matrix implementation, which is usually an error matrix that is used to summarize the results of a performance evaluation when the true values are known. So in essence, also our approach is to evaluate any method such that the retention and RRF should be calibrated. And after our calibration, you should define first the limits of quantitation. And my apologies, I went a little bit ahead. So in general, you should define first the limits of quantitation. And second, it's to predict the RRF and the retention index in the universe above and below those limits of quantitation. Apologies, my mouse keeps scrolling up and down from this particular slide. So, and third would be to analyze and determine the accuracy of the predictions and repeat experimentation under various instruments and column conditions. And once calibrations conditions are actually determined. So our intention is to then develop a scorecard, as I previously mentioned, to show the boundaries of the analytical method and determine the detectable and undetectable space of that breadth universe. So from our ongoing work, we have actually developed an initial framework with the intention on how you can develop your method and process your data. The first step involves establishing your retention windows and calibration parameters. For these steps, we've developed a simple QSAR model based on semi-empirical approaches, and we've also implemented the use of Abraham coefficients that relate to the column stationary phase. And all of these are very method specific, so that's usually the first step of this particular method development as part of the data processing. So for the second step would be a series of steps that you can analyze your response ladder in general. And then as part of your third step would be to define a detectability range. So this step usually involves the determination if the compounds predicted within a particular retention time and response are within a particular detectability range. So so currently, as part of some of our initial results, and we pretty much have taken the approach of from the stepwise approach as discussed in the previous slide, we have been able to develop some early models based on the concepts of the electron ionization cross-section concepts. So we've been able to show some linearity across some of the different functionalities of the chemicals that we've so far analyzed. And through our modeling in general, we've been able to analyze at least 80 chemicals detected in our laboratory. With our simple Python modeling, along with our the molecular descriptors that we've selected for this particular work, we have been able to work with different test and calibration sets as well. And we've been able to build multiple linear regression models that in general best describe a linear response, as you could see on the bottom left graph of our experimental data. So this specific model, and in general, we don't have all of the 80 chemicals. We've included at least 50 different data points showing the calculated experimental relative response factors versus the predicted relative response factors. So at this particular stage of our experimental process, a lot of work still needs to be done. As you can see, we need to still improve some of the R values for this particular methods that we're sort of developing. But our intended next steps are to sort of keep evaluating that method performance and combining all the right molecular descriptors to predict the retention index and the detection capabilities of the method. So we also want to reduce the dimensionality of the data to create a map, as you could see on the right-hand side of the slide. So we want to create this map of a chemical universe to chart all of the laboratory performance into what we call the chemical space. So as I've also mentioned briefly, as part of the anticipated final results, once we have determined all the specific breath compounds from our universe or from our work, our universe will consist to evaluate several different GCMS methods, employing a non-targeted approach. And when we are sort of evaluating our GCMS methods, we will be considering all the modeling and the prediction, retention of all the analytes in the breath universe, as well as all the response factors of such analytes. So both the retention and response predictions are combined, and it's expected to determine the detectability of the space within that particular universe. So as an example, I presented here a scorecard in which will be prepared, in which it actually does intend to show the boundaries of a chosen analytical method of the detectable and undetectable space of our breath universe. So furthermore, that particular scorecard is expected to show inter-laboratory comparisons and how well that laboratory performed using that specific analytical method that we've provided. So in general, this concludes my presentation. But before, I want to thank everybody here at the FDA, also on DBCMS, and also the Bill and Melinda Gates Foundation, as well as Owlstone Medical. And thank you all for listening to me today. Thank you all.
Thank you again so much for taking the time to run through the presentation. Quick reminder to everyone, post the questions in the Q&A on the right-hand side. So first question here is probably more for Victoria. I think a lot of people that are more involved just on the pure research side of things. I was wondering if you could explain further in layman's terms, what are regulatory sciences tools and why are they important for those different types of users?
Yeah, so that's why I tried very hard in my presentation to cover it, but if you haven't used it before, it could be very difficult in first pass to understand what they are. Regulatory science tools could be, they are simply a, in our area, let's pick an example. In analytical chemistry, they can be a recipe for how to do an experiment and how to analyze it. It could be an analytical method, which would include sample preparation, the analytical method, and how would you set up your system, what would be your parameters, and it would guide you in a way how to do an experiment. It becomes very important when you are looking at an area where maybe the ISO is not covering it. Maybe there is a gap in the standard and you need some understanding how to do a certain experimentation. Therefore, the RSTs are aimed to address those gaps and teach you how to do the experiment and get the data in support of your submission. That's the aim that we have with the RSTs. Ultimately, what we would like to do is to either help close the gaps in the ISOs or actually develop new standards for areas that standards are not available. And these RSTs in combination with the publications can actually do that. The next thing that I think is very important for the folks who are on the call today at this conference, that to know that we take feedback on our regulatory science tools. So if you download a tool and you're using it and you find something that you think it's very important for us to know, please, there is a feedback section on that tool. provide your feedback, because we go in and we check to see how we can improve these tools. So for example, if you ran a method and you got a little bit of a different answer that is concerning, we would like to know that. We also look at the traffic. If a tool is receiving a lot of traffic, that means that we are moving in the right direction. If a tool is not getting a lot of traffic, then that means that maybe we're not doing things right. Maybe we should go back and think why this tool is not receiving the attention it should. So in layman terms, a tool can do a lot of things, and we are hoping to be able to allow for those companies that are maybe younger, maybe they don't have the means of having a huge regulatory group, to use these tools to be able to cut down in the process and help them, you know, with their technology development. That's our goal.
Fantastic, fantastic. Next one, I think probably for you as well, Victoria. Could you comment on an example of a regulatory science tool in the catalog? I know you shared a screenshot and there was a link there, but I was wondering if you could elaborate a little bit more on maybe an example.
So there are a number of examples in our group, the chemical characterization group. Our most recent tool, and I think Billy, you and I have talked about this before, our most recent tool that went on the catalog was May of 2024. It's a tool that has looked at 107 reference standards for materials that goes into medical devices. And we are trying to, our aim is to develop a coverage map for these materials that are used in medical devices. And so this tool is very important because it will tell you, okay, if you are doing a submission, you may want to take a look at this and evaluate your material. The tool is so important that within the first day, the hit on the tool was 4,000 people actually went in and took a look and downloaded. That tells you how important the area is. And we're actually continuing. The next tool that is related to medical device reference standard material is going to come out. We're starting to work on that, and we are going to cover a number of materials. So one would be additives, the next one will be polymers, oligomers, and monomers. The next one will be, colorants. There are, we take all of that into consideration, and a lot of work went into getting the tool together and put it, because we had to look at a lot of chemicals and consider whether these are actually ending up in a device and then manage it from that. There's a whole team that works on that. They're not here today. Omar was partially a part of that team for a while until he moved into the breath work. But yeah, that's an example of a tool that we have recently put on our catalog. 2 more is in the process of coming. As soon as our publications are approved by the journal, the tool package goes into our tool team and they look and review. It gets reviewed, and once we go through the corrections that they ask us to do, and it's approved, and then it goes on our catalog and it's available to the public for free to use it. It's open to public. So that's how it works.
That's great. I think it's interesting because you can see how there's read across in chemical characterization. There are 2 similar types of challenges that exist in the breath space. So it's a wonderful foundation for a lot of the challenges that the breath community face. Next question, maybe for Eric, it kind of relates to some of the challenges within the breath field. You mentioned the point that getting chemical standards, it's expensive, it's hard for small groups to do, etc. I was wondering if you could elaborate a little bit further on what you think some of the historical challenges may have been for breath, and how some of these approaches around confirming the ID, ensuring something is on breath, may help in the future.
Absolutely. Great question. Historically, the challenges were, I'd say, sample collection, capturing all these volatiles without background contamination has and continues to be a huge challenge. Instrumentally, we've come a long way. Now we have high-res instruments, many different kinds to help confirm that identification. But historically, I think it's been sample collection and high-resolution mass spec, so quality of data issues. Now I think with many different manufacturers making great high-resolution mass spectrometers, and then also GC technology is, expanding with, 2D GC with the ability to separate so many volatiles in a sample. I think that's contributing to our ability to identify many of these trace level analytes in the breath. So I think moving forward, we'll see a lot of confident identification coming out in the literature. I think streamlining the workflow will really help labs communicate and verify identifications so that the whole breath community can work together and move forward and then do these type of quantitative analyses.
Yeah, no, that's great. And definitely you can see, as you say, with the different technologies becoming available, if you know what the compounds are because you have a confirmed ID in the concentration, that does help you use all of these different analytical platforms and then have comparability across data as well, which I think is going to be fantastically exciting for the field. Next question here is probably for Omar. It's kind of two related ones. So except Python, are there any other tools such as AI that could be used to predict retention?
Yeah, absolutely. There's plenty of more sort of particular tools used to predict the retention in general. We've explored several different techniques in general when we were sort of in the initial stages of this particular study. So in some of the studies that we've actually have modeled, some of our semi-empirical approaches do take an AI and in essence also sort of like a machine learning type of techniques in general. We're not using all of those very complicated particular type of modeling because one of the pitfalls that you may fall into is that you have to generate an insurmountable amount of data in order for those particular models in essence to sort of work properly. So on our end, that's why we sort of have taken a sort of a semi-empirical approach, and we're not using any sort of complicated, you could say quantum chemistry sort of programming, just because in essence, we're sort of trying to test all these particular methods without generating a high level of or generating a high level of data. So yeah, there's a lot of techniques out there. One of the ones that are that we also kind of try to model a lot of our things were from neural networks in general. So there's plenty of techniques out there that you can definitely model all of this based out of. It's actually what you use, what works better for your actual type of application.
Yeah. Next kind of follow on to that, and this is one I was thinking as well. Is there effort to include other mass spec methods other than GCMS, specifically without GC? So I know a lot of the breath research community will use SIFT-MS or PTR-MS. Do you think there's kind of scope to use these same types of techniques for those other analytical instruments?
I don't know if that's directed at me, but at least
that's at you. I would imagine that one.
So I would not for a particular project that we're doing currently for breath. It's another project that we're doing here internally. We're not just focusing completely on using GC techniques. We're focusing on LC techniques, but as far as what I'm currently doing, I haven't explored any sort of other techniques that sort of don't employ GC. So that's something that definitely I could discuss within my actual work group to see if there's any other sort of techniques we can implement to this type of research. I'm pretty sure there's plenty of other mass spec techniques that we can apply to what we're currently doing.
Fantastic. Next one, I think, is probably for Eric. So you talked about the low res and the high res instrument in the talk, and you outlined our things like GC-GC. Is there a plan to do GC-GC, or will it be using different column chemistries in your analytical work, Eric?
Our short-term goals is to use different column chemistries and to continue our current work looking at different stationary phases. Yes, but GC-GC is not off the table. In the short term, we will have 1D GC, high resolution. But I do foresee, because it's a very popular technique in the breath area, I do see tandem GC in the future. However, I cannot say how soon that will be. But we have an excellent lab here, and we're always expanding our instrumentation. So it's something we've been thinking about.
Excellent, excellent. Thank you. And again, I suppose if different groups are IDing and quantitating, then we can coalesce different data sets together as a whole field. The next question is for you as well, Eric. Is NIST library the only reference library being used currently?
Great question. No, we're not just using NIST. We're also using Wiley, Agilent, and other resources out there. We prefer to use publicly available databases to more accurately pull from readily available sources and not favor any commercial databases. So we're looking at as many sources as we possibly can. NIST is the most used, and so we thought that would be the best place to start.
Fantastic. Next one, this is for Omar. Fantastic talks for Omar. Do you think a similar modeling approach could be used for prediction of pre-analytical sampling efficiency, e.g. for prediction of recoveries of on-breath VOCs using different sorbent types?
Oh, that's a fantastic question in general. I was seeing it here in the QA. We really haven't discovered that particular part of a pre-analytical sampling efficiency. That would be also one of those particular topics that we could look more into in general. That's where, and for this particular talks, that's why I take some info in general. So that's one of the topics that we can also sort of dig into for future projects. So as far as in general, for different sorbent types, I don't know if the person that asked the question means different sort of columns that we're using for this type of work. If it's more related with the columns, then yes, at least for my ongoing work or future work, I would say within the next couple of weeks, I'm actually going to be trying all of these experiments that we've designed in the laboratory, and we're going to be applying pretty much everything to different types of columns. Analytical columns. So some of the reasons being is that a lot of our modeling also takes into account the packing inside of the column in general, their stationary phase. So we're sort of designing all of these experiments in the laboratory so it could well align as well with a lot of our computational modeling. So hopefully that's what the question was referring to, more like the column packing, hopefully.
Thanks, Omar. Time for two very quick ones. This is for you outside Victoria, and this is something that we've talked about before as well. VOCs, not just in breath, but if you look in the head space of other samples, like a blood sample or urine, you would expect that, you know, that's fundamentally where the VOCs are coming from. Is there plans to look at other sources of VOCs from these other sample types?
Yes, and there are a few things I'm going to cover for plans in the future. Obviously, what Eric and Omar and the team have presented is just the beginning of our work, Billy. I think, and we're focusing predominantly on GCMS because that's where we think that the start has been to develop for breath. You know, the studies are certainly whatever is in literature and what we have found. And if we are going to build the base, you know, you should start where there is some data so that you can take a look. And predominantly, I think GCMS has been something that they have looked, whether it's Orbitrap or something else. Plan for other methods, possibly. You know, I think once we get our handle on this confident identification using GCMS, then if there is a potential for other methods, then we can address that. That would be probably the next level, I would say. I think it's important for us to build the base and then kind of move over. Plan for other areas. Yes, other body fluids are very important because I think it will simply add to the confidence of the breath. If you could correlate, if you could do the work and then correlate the biomarkers that you see in breath with other body fluids, I think that would be tremendous because then it would add to the you know, evidence that one needs for the work. And we have plans to do that. And we are thinking about it. We've just started this work and the group has been moving tremendous. I mean, these guys are every day they're doing something. And we started this in April. And since April, we've come, you know, quite a bit in an area that I have to tell you, none of us were an expert. And just by studying and working and talking and learning, we've brought ourselves to this level. And of course, we are analytical chemists by background, but we had some, you know, steep learning. We are hoping to use TB as a base because it's a disease that possibly it's easier to look at, and there's a lot of literature out there that one can rely on, and hopefully we can cross-check this biomarker to get this confident identification. We're all seeing the same thing. And then we would like, if possible, for us to move to the next and the next and the next. So that's our plan. And that's what we're aiming for and hoping that we're intending in the future to expand to other body fluids and expand into other diseases as well.
Fantastic, very exciting. And you mentioned earlier that, you know, always wanting feedback. I love this last question of the session. It's how can members of the breath community help in this effort?
A lot. They can do a lot. So, you know, we're always open to working together if there is a possibility. If there is a situation where, you know, someone is using another piece of instrumentation that we don't know about, it would be nice to know about. I think collaborating with the breath community, all of us together can bring that confidence that we need in this area, in this arena. And feedback, however you can add value to this and share, I think it's going to make us better at what we're doing.
Wonderful. And on that note, I want to say a final thank you for all the contributions today. It's been really exciting and we're really appreciative of you joining us today.
Thank you. Thank you. It's a pleasure. Thank you for having us.